Hello everyone. How are you all? Hope all are safe at home. Welcome back to biology class. Today's topic is antibiotics. What are antibiotics? Antibiotics are substances derived from a microorganisms or produced synthetically that destroys or limits the growth of a living organism, usually pathogens. An antibiotic is a naturally occurring, semi-synthetic or synthetic type of agent that destroys or inhibits growth of microorganisms. And the kinds are natural, semi-synthetic and synthetic. Natural ones are mainly from the fungal sources. Semi-synthetic, chemically altered natural compound. Then synthetic, chemically designed in the lab. Then antibiotics are classified in several ways on the basis of mechanism of action, on the basis of spectrum of activity, on the basis of mode of action. Actually, the classification and the type of antibiotics are not in our syllabus, but these are for your further information. So, on basis of the mechanism of action, antibiotics are classified as following: protein synthesis inhibitor, cell wall synthesis inhibitor, DNA synthesis inhibitor, RNA synthesis inhibitor, folic acid inhibitor, and mycolic acid synthesis inhibitor. I need to by heart in our syllabus. Then, on the basis of mechanism of action, cell wall synthesis inhibitors, examples are here. Then, protein synthesis, another examples are here. Then, DNA synthesis examples, RNA synthesis. Then, folic acid, mycolic acid, examples with the names, chemical names are there. On the basis of spectrum of activity, these are classified into broad spectrum antibiotics and narrow spectrum antibiotics. In the slide, it's written short spectrum. These two terms are there in the textbook. While reading the textbook, you can go through it. On the basis of spectrum of activity, antibiotics are classified as broad spectrum antibiotics. The term broad spectrum refers to an antibiotic that acts against a wide range of disease-causing bacteria. Examples are here: tetracycline, amoxicillin, etc. Then, short or narrow spectrum antibiotics. The term short or narrow spectrum antibiotics refers to an antibiotic that acts against a narrow range of disease causing bacteria. Here comes the examples: penicillin, then vancomycin, etc. Then, on the basis of mode of action, bacteriostatic, it is an agent in biological or chemical agent that stops bacteria from reproducing while not necessarily killing them otherwise. They are just stopping them from reproducing. Then bactericidal. Then it is an agent which or chemical agent that kills the bacteria. Then here comes our terms. Resistance. Resistance means, you know the word meaning of resistance. If the concentration of a drug required to inhibit or kill the microorganism is great or less than the normal use, then the microorganism is considered to be resistant to the drug. Cross resistance, no need. How do antibiotics work against bacteria? You know, antibiotics can destroy bacteria, but it is not effective on viruses. So, how it works against bacteria? Antibiotics fight bacterial infection either by killing bacteria or slowing and suspending its growth. And how it is possible? By attacking the wall or coating surrounding the bacteria, and by interfering with bacterial reproduction, and by blocking protein production in bacterial cell. Then, antibiotic resistance occurs when bacteria can no longer be controlled or killed by certain antibiotics. In some cases, this can mean there are no effective treatments for certain conditions. Why antibiotics don't work against viruses or less effective to viruses? Antibiotics cannot kill viruses because viruses have different structures and replicate in different way than bacteria. So as you know, viruses are non-living particles. Antibiotics work by targeting the growth machinery in bacteria to kill or inhibit those particular bacteria, but it is not effective on viruses. Then, When you think about it structurally, it makes the sense that an antibiotic could not work to kill virus with a completely different set of replicating machinery system in it. Four ways antibiotics affects bacterial cell. You can see the ways here. By disrupting the cell wall synthesis, by inhibiting the RNA synthesis, then by inhibiting the DNA replication, by inhibiting protein synthesis in a bacterial cell. So just learn the names only or the ways only. Mechanism of antibiotic resistance. There are a number of mechanisms that bacterial cell use throughout to effort of antibiotics. Some bacteria are naturally resistant due to an unusual impermeable cell membrane or a lack of target that the antibiotic attacks. So some other bacteria are capable of producing enzymes that can inactivate antibiotic upon contact. In some other cases, bacteria can modify the antibiotic's target, rendering the antibiotic ineffective. Bacteria can also produce efflux pumps, which can transport antibiotics out of the bacterial cell, therefore, they can take effect on it. Then, what are the ways by which bacteria acquire resistance? You can see here resistant bacteria is by mutation and horizontal gene transfer. So, two main reasons for the bacterial resistance: mutation and horizontal gene transfer. So, here comes. So, take your own time and read it. As the video is getting more long, I cannot upload it. Okay, so I am concluding the points. So, what are the consequences of antibiotic resistance? Treatment failure leading to chronic problems. Then, increases spread of antibiotic resistant bacteria and their associated community related problems. Then, relapses of infection after the treatment also. Then, increased mortality. Then, adverse effect of alternative treatments. Then, lack of availability of clinically effective antibiotics, longer and more complicated hospital cases, excess healthcare costs. These are some consequences of antibiotic resistance by the bacteria. Then, how to reduce the spread of antibiotic resistance? Do not use antibiotics to treat viral infections such as influenza or common cold. Then, take the prescription from the doctor only. Use antibiotics only when a doctor prescribes them. When you are prescribed antibiotics, take a full course. Don't stop in between and don't take over course. Never share antibiotics with others. Then remember, each time you take an antibiotic when it is not necessary, the effectiveness of the antibiotic decreases and it might not work the next time when we need really it. Then here comes the end of the topic. As I concluded the topic in a fast way, you can read pages 213, 214 and 215 from the textbook. Okay, so mainly go through the reasons of bacterial resistance, then mechanism, read it carefully. Then here comes the ways: mutation and horizontal gene transfer. Then consequences and how to reduce the antibiotic resistance. Okay, so we'll meet you with another video and have a nice time. Thank you.